Hello everyone, how are you? Thank you, Nova Nutrients. Thank you for joining us today. Hello everyone,
It's
how
great to
are
be
you?
able to share our work
Thank
with
you,
you and
Nova Nutrients.
hopefully
Thank you for
learn more about what you're up
joining
to
us
these days.
today. Hello everyone,
It's
how
great to
are
be
you,
able
Pablo? Yes, it
share
was
our
a
work with
really
you
fast-paced
and
presentation. I
hopefully
hope that
learn more about what you're up
joining
to.
us
As I
these
said,
days
the
today.
level of technical detail and
Hello
amount
everyone,
of things
how
ready to
are you,
cover
Pablo? Yes, it was a really fast-paced presentation. I hope that you're proud of what we have achieved this
level of
year.
technical detail and
Hello
amount
everyone,
of things
how
ready to
are you,
cover.
Pablo? Yes, it was a really fast-paced presentation. I hope that you're proud of what we have achieved this
level of
year.
technical detail and
So we wanted to make sure that we were showing the presentation. I hope that you're proud of use this as we have achieved this
level of
year.
technical detail and
So we wanted to make sure that we were showing the How about now? It has improved now? Great. That's great. I just wanted to make sure that nobody was going deaf, so I wanted your feedback. Well, that's great. So I was mentioning we're really happy of being able to share what we've done during the last 18 months with you, and we're looking forward to unlock some of the milestones that we showed today. So we felt like sharing those achievements with the community and start focusing on next milestones. We presented the line of 3D printers today. That's going to be super exciting. Keep scaling those systems and also increasing the capabilities of the caster, as well as start working with our biopharmaceutical partners, starting to have engineering reports about the performance of our technology with their cell lines, and hopefully start installing some of our first prototypes inside GMP facilities to test the integration of our technology with current manufacturing pipelines. So the aim of these 20 minutes, 30 minutes that we are opening here was to create a channel of conversation in which you can ask some questions regarding what you saw, what did spark your curiosity, and hopefully have an opportunity to expand a little bit on that. Oh yes, so thank you for the support team at Stämm that is sending some of the questions asked during the demo day. So the question is if the bioprocessor can be operated only at a lab. So today the prototype that we are working with require a step that they need to be managed under a hood. The industrial design of the bioprocessor, and specifically the strain on a chip, is intended to remove the necessity to manipulate cell banks and manipulate cell lines to reactivate them. So ideally, when the product is finished, you wouldn't need a safety hood in order to manipulate these cell lines and have a continuous growth of this particular strain that you want to work with. So I would say that the main reason that we started with this concept was because we were faced with the hardship of getting your hands on all the equipment and all the infrastructure required to perform bioprocesses. So we wanted to remove that from the equation, and I know it's a big challenge. We love working on that, and we believe that with microfluidics and laminar flow approach, we can achieve an isolated environment from the and sterile environment that actually always is pumping up out cells. And for mammalian cells and maybe pharmaceutical applications, that's even a little bit more regulated, and there are some options that won't apply for operating this machine outside a safety environment. But there are other applications where the requirements and the regulations don't or the application is not linked to a safety or not safe, but a safety hood environment or ABAC environment. Okay, so the next question is, can photosynthetic microorganisms be used in this device? That's a great question. We definitely the work that we show today is not focused around photosynthetic organisms. We always have look at that application dearly, and we have some approaches in mind on how to enable a homogeneous access of light to the cells inside the system. So hopefully that's something that we would really like. We love vegetable cells, so we look forward to the time in which we can also grow vegetable cells in our lab. Okay, so Micah, yes, he's asking regarding the impact of shared forces in mammalian cell culture against yeast and bacteria, and that's a fair point in terms of when you look at high scale production. Part of what we've seen is that when you look at the ceiling of a scale that we have reached in the mammalian cell culture industry, that is tied to shear forces. That's not the case for yeast and bacteria, and also the efficiency of conversion of the media for bacteria is really good. In those cases, we believe that the answer is the same regarding the capability of implementing these bioprocesses in a decentralized manner and enabling more people to think about bioprocesses as a solution and as a design tool bespokely customized for the resources that are locally available. So I think that if we keep intensifying centralization, we also will keep intensifying the sources of carbon that we're going to use for those bioprocesses, and also we're going to restrict a lot the access of to that technology to a really few group of people. So I think that the more leverage that we can do on biomass, local biomass, to replace local supply chains with bio-based materials, that's a good step for start scaling locally to globally. And hopefully by the time that process happens, the technology also will be in a stage that can follow that growth and provide the infrastructure required for production in those kind of cultures. Yes, this is from GridX. Disclaimer: GridX is an investor in Stämm and very good friends of the company from the beginning. And we also love to dream about. We also have this idea of if you design for space, it's probably going to be really easy to implement that on Earth. So we love to say that microfluidics channels are really suited for microgravity, basically because you delete that effect of sedimentation that we were talking in the demo day, and that gives you a really predictable path of the cells and also a really small footprint. And those are the things that are scarce in space. Yes, so the next question is regarding the expertise or the kind of tutorial that you need to go through in order to operate a bioprocessor. Hopefully this process is going to be as automated as possible. The parameters that you have to adjust in the bioprocessor are fairly few. I would say that the most significant are the formulation of the media and the feeding rate and flow velocities that you're choosing, and also the formulation of gases in real time. So I think that hopefully we can allow the user to set thresholds of work and allow the firmware control to manipulate the feeding rate and the formulation of media and formulation of gases to keep the bioprocess within those thresholds. Those thresholds will be things like cell density, pH, some levels of certain sugars, or dissolved oxygen. Yes. Yes, definitely. So that's a good question. For applications like mammalian cell culture, we are thinking of single-use systems. So you have a hardware scaffold that is not single-use. That is where basically the stereo-on-a-chip, the bioreactor on a chip, and the biofuel bioreactor as a closed and connected system is plugged in, and specifically for the production of biopharmaceuticals, you could use that system for several months. But once that process is done, you have to change the 3 microfluidic devices. On contrast, to that is if there in an event of a contamination, I would say that there is a benefit on the fact that the amount of total volume inside the continuous system is lower than a large system or a single-use bag system. So if you have a 10,000 liter bioreactor and you detect a contamination, all the batch is compromised. So I would say that here you as you are sampling in real time and you are collecting a library of biologic samples through time, you can release a partial production that hasn't been in contact with a potential future contamination. So definitely the concept of batch for something like this has to be revisited and adapted, and also the release of partial batch as well. Question of Lucas: what are the first steps for Stämm in the biotech market? Well, we were mentioning now, by the end of the demo day, the main focus of this year is to keep developing our commercial relationships with biopharmaceutical companies and CDMOs that basically are looking at future generations of the production of antibodies, and start doing all the way into the industry, being really diligent, starting by doing engineering runs at a R&D scale. After that, installing a pilot prototype inside their facilities and develop the installation protocol, the performance protocol on site, and develop insights regarding how this technology integrates with their tools, and hopefully start funneling new molecules through clinical trials with our technology in a way that is suitable for scaling to commercialization with a percentage of their portfolio. So we're really happy on the success of this first demo day. For us, was a really good experience to expose us to share our work, and we are happy that we have more than 100 attendees. Hopefully we will put this event also available publicly in the future days, and you will be able to revisit it or share it with friends. We have another question. Will it be possible for people to use the bioprocessor at home at some point in the future? So yes, I mean, like I would love that. I think that we are in an industry, as I was saying, that is highly regulated. There's a lot of safety concerns that we must address in every step of the way. We believe that biotech is a transformational technology, and given the challenge that we're facing, we need more people thinking in terms of biotech as a tool for solving local and global challenges. So how you produce a solution is a structural part of being able to implement a solution, and for biotech, that is bioprocessing. So hopefully in a safe display, and that's something that can happen. We talk about a lot of things today in all the steps on developing the biomaterials. That was super exciting work in terms of what can be done for functionalizing those biomaterials and bringing in new possibilities also to the industry in terms of how we implement treatments inside the bioreactors. We believe that with the upcoming of cell therapies and all these protocols that require training of cells and protocols of differentiation with several stages, having a way of drawing a line in space and being able to deliver differential treatments in time can add possibilities that aren't available today, and can make possible that some treatments are more scalable or easier to scale at a larger scale than they are today. And then the caster, that's huge. We are nerds in the sense of that we love the work that we do, and we understand what it took to develop that software and how significant is the increase of power that we can have in designing microvascular systems. So that's why we are so proud of it, and we want to make a software and a product line out of it. So I don't want to talk alone. I don't know if someone else has any other questions or maybe ideas, suggestions. What aspects of our company would you like us to cover in our next demo day? Or also we can decide to stop. You let me know if you would like to look into something else or. Okay, I think that we're good. I thank you very much for joining us today. We have shared our contact information. And let me say it again. You can reach out to us at info@stämm.bio for further details, and also if you are interested on particular applications and opportunities. This is the time. This is going to be a year in which we are specifically focusing on testing this technology for different applications. So we encourage you to reach out to us and design together an action plan to start building the bioprocessing industry, the remaining 99%. So thank you very much for joining us, for taking the time, for learning more about our work, and we hope to see you in our next demo day at some point in the future. Have a great rest of the day. Take care.